guys, I think we can start now. Okay, welcome to week 9 for the live session of this course, Bioinformatics Algorithms and Applications. Today we are going to talk about protein folding primarily, different types, like different factors affecting protein folding, different rates of protein folding, and specific uh, protein folding for different class of proteins. And we're going to also touch a little bit about CAD, computer assisted drug designing. Uh, so I'm not going to see any demos in this session, but like I'm going to discuss the overall concepts and yeah. Okay, so let's get started. So the first concept of comes from protein folding rate, right? So I hope all of you can hear me. Please let me know if my voice is not audible. There's some background noise here as well. Okay, first we're going to talk about protein folding rate, right? So we have to,
Right.
yeah.
Oh, that's the reality, bro. They isolate heat, they enjoy.
Sorry, I can't hear you. Can you speak a little louder?
That Nitish Nitish also studied here only, no? Reform. Bro, Nitish is goodbye, bro.
Hello.
Naveen is toxic. Wait, I will say to Naveen tomorrow. The captain said like this. Hey, ma'am,
Okay, I'm
send one
muting, I
message.
think,
Sparkle
Ranveer for some
kind
reason.
contest.
Okay, please, if there's some technical issue or my voice is not audible or anything, please let me know. Okay. Okay, so yeah, we have discussed about different secondary structures earlier, even a little bit about tertiary and quaternary structures. There are different bonds that give stability, different chemical interactions, different ionic interactions, even physical interactions like van der Waals forces give stability to the whole secondary, tertiary, and quaternary structures of the proteins. Uh, but we are going to talk today about the process of folding of the protein, like how is the because to get to the secondary and the tertiary structure, the protein needs to be folded in some manner, be it in an alpha helix or a beta sheet or any kind of tertiary structure. It needs to be folded upon itself, and as I was saying, several chemical and physical interactions govern this folding, and most importantly, it is also governed by the sequence of the amino acids, right? So if you want to measure the folding rate as in how many folds per second, right? Like the once the sequence of the amino acid is formed from the mRNA, if you have just the primary sequence, what is the rate of the folding of the protein per second, right? How many folds per second is the rate, right? So it is a measure of the speed at which protein transitions from an unfolded to its native 3D structure. Uh, why do we need to study protein folding? It is one of the major questions that you should have an intuitive answer to before studying protein folding, right? So why do we need to study protein folding? Because misformed folding of the protein or misfolding of the protein or like not correct folding of the protein can lead to a lot of things, right? So the structure of the protein, as we have discussed earlier, for say for example for enzymes, the structure, or for even for other proteins, the structure of the protein is very very important, right? It is what determines how it is going to function. If it is an enzyme, the structure needs to have an active site where the substrate is going to come and bind, and then it can do the chemical reaction and so on and so forth. If it is a transcription factor, it needs to have binding ability to DNA. So the structure needs to be like that so that it can go and bind to DNA. So generally, the structure will determine how the protein will function, and on the overall, the structure is determined by the sequence of the protein, right? And misfolding of these proteins can lead to different diseases like Alzheimer's and Prions diseases. And what are the level what are the levels at which we study the protein folding? First is we study the chemical interactions. We study what are the chemical interactions that are helping this protein fold, or why is the protein folding in a certain way because of those interactions. Second is like what kind of disease can be formed because of differential folding. And third is basically we also can understand protein origins and evolution of the protein uh, through the folding of the protein. So if you have two proteins which fold similarly at a similar rate but different kind of they have different mutations. If you compare them side by side, you will find certain regions have been mutated. It means that in an evolutionary perspective, that protein has been evolved from, say, for example, from chimpanzee to humans. The folding has remained almost similar, so the functionality remains the same, but so certain residues has been changed or mutated or something like that. So three levels we study the protein folding. First is at the chemical interactions level, what kind of interactions this does help the protein to fold properly, what kind of disease is formed when the protein folding does not happen properly, and what evolutionary insights can we get comparing protein folding of different of say, the same protein across different species, right? Now, a little bit about the diseases that are caused. The most common example of the disease that is formed by differential protein folding or misfolding of proteins is the Alzheimer's disease. So, this is one of the major neurodegenerative diseases, and the prevalence of Alzheimer's disease is very high currently in low middle income countries like India, where we have a vast population and a significant portion of this population are above a certain age who can develop Alzheimer's disease. So, diseases like Alzheimer's disease is is very impactful to the society as a whole, and also we do not exactly have exact clinical treatments for diseases like that. But Alzheimer's disease is primarily formed. It's a complex disorder. Like there are several symptoms of the disease, but the primary cause of the disease is misfolding of the beta amyloid protein. Right. So the amyloid beta protein or the beta amyloid protein is one of the proteins that is present in different neural cells of the human brain, and misfolding of this protein can uh, misfolding of this protein can lead to accumulation of misfolded proteins inside the cell, which can lead to this disease. So, senile plaques are formed because of these accumulation of these misfolded proteins. They contain aggregates of the protein beta amyloid because this is not folded properly, it is not being able to function, and also it is not being able to be cleared from the cell because of certain issues as well. So these keep on accumulating. The misfolded protein keeps on accumulating within the cell, and the level of beta amyloid increases a lot. Right? Right? So there is another polypeptide produced by the proteolytic cleavage of a larger protein called as APP or amyloid precursor proteins, and so this protein, this beta amyloid is formed from APP, and this beta amyloid is there is no way to clear the beta amyloid from the cell, so it gets accumulated, and it undergoes a conformational transformation to form a soluble alpha helix to a beta sheet, and it's prone to self aggregation, as I was saying. And ApoE or apolipoprotein E is a potential mediator of this conformational transformation. So this is the marker of hallmark marker of Alzheimer's disease is misfolded proteins getting accumulated inside the cell, and there is no way of clearing it, which increases the level of a certain protein, which leads to the disease. So that is Alzheimer's. The second is beta thalassemia. Beta thalassemia is also very common in certain Indian populations, uh, where it is basically uh, caused by mutations in the HBB gene, which is the hemoglobin gene. So it, it is caused by genetic defects that impair synthesis of one of the polypeptide subunits of the hemoglobin protein. And during the burst of hemoglobin synthesis, a specific chaperone called hemoglobin stabilizing protein or ASHP. HSP is basically heat shocks proteins or other type of chaperone proteins that are involved in the folding of other proteins. So for hemoglobin, this chaperone is called hemoglobin stabilizing protein. It binds to the free hemoglobin alpha subunits, awaiting the incorporation into the hemoglobin multimer. In the absence of this protein, a alpha hemoglobin subunits aggregate, resulting in precipitate that has cytotoxic effects on the developing erythrocyte. So it is the misfolding of this protein that is becoming dangerous, and that is accumulating and becoming dangerous to the development of the cell itself. So these examples are to tell you that protein folding can directly cause diseases, right? Misfolding of proteins can directly cause diseases. Uh, but in a lot of cases, we do not see outcomes of misfold, because in maximum cases there are mechanisms within the cell. If a protein is malformed, or misfolding is happening inside the protein, there are mechanisms which can take the protein and uh, cleave the protein and destroy the protein and throw it out of the cell. So in most cases, the repair mechanisms work, but in certain cases, like as I was saying, Alzheimer's disease, there is no way to clear the amyloid beta accumulations, which can lead to the disease, right? Now, now we are talking about protein folding rate, right? So we know that the protein is folding at a certain speed. We want to quantify the speed in some way possible, right? So it can, again, if you look at a host of proteins, it can vary, and technically it varies across 8 orders of magnitude, right? So I think this is the streptococcal protein folds the fastest, as at 10 to the power minus 5 folds per second. Tryptophan synthesis is a protein which kind of folds the slowest, which is like 10 to the power negative 3 folds per second. So uh, but why do we need to know this, right? The question has stimulated experimentalists to seek fast folding proteins and to engineer them to fold even faster. Proteins folding at or near the speed limit are prime candidates for all atom molecular dynamic simulations. So if you're doing any kind of MD simulation exercise, fast folding proteins are the ones to go for, right? They may also have no free energy barrier, allowing direct observation of intermediate structures on the pathways from the unfolded to the folded state. Both experimental and theoretical approaches predict a speed limit of approximately n by 100 milliseconds for a generic n residue single domain protein, uh, with uh, with proteins folding faster than B or AB, right? The predicted limits suggest that most ultra fast folding proteins can be engineered to fold even faster, right? So the the pace of the speed at which the protein folds is of interest, because you can use that speed to engineer some proteins. So this comes very handy in cases of drug design experiments as well, right? Now we talk about what are the factors, what are the interactions that are driving the folding of the protein, right? So the major ones, as we have discussed earlier as well, are the hydrophobic interactions. The hydrophobic interactions are very important, because that will decide which way the protein will fold, because the hydrophobic in sorry, the hydrophobic residues and the interactions of those hydrophobic residues are always going to be buried well within inside the protein, right? So the hydrophobic interactions will determine which side the protein will fold to. So this is the most dominant force. The protein adopts a conformation in which the hydrophobic amino acid tend to be buried inside the interior of the protein. The second important bonding is the hydrogen bonding, in which the atoms along the backbone and sometimes also in the side chains try to form hydrogen bonds with each other. So as we have discussed earlier, the hydrogen bond is formed between the carbonyl and another hydrogen atom of any other residue, right? So if you have so this also quantifies to long range contacts as well. So if you have a folded state, you have two amino acids which would generally not be very close to each other, but they are close to each other, and they are polar in nature, so they can form hydrogen bonds which will stabilize the structure of the protein. And then there are other interactions which are weaker, for example van der Waals interactions. To drive a compact structure from hydrophobic interactions promotes favorable close packing between protein atoms. And there are also electrostatic interactions, because there will be polar amino acids. Basic will form bonds, electrostatic bonds, or salt bridges, if you remember, with basic will form with acidic amino acids. Now the question comes, will if you create a random sequence of amino acids, Will it fold like a normal protein? The answer is no. People have tried to do that. If you just put a random sequence of amino acids and you try to fold the amino acid, it is not going to fold. So there comes the idea that nature has selected the sequences that are able to fold among other things, right? So the sequence selection, the selection pressure that is acting on the protein sequence that we have discussed earlier about multiple sequence alignment, you tend to see certain residues getting conserved, certain residues that can be mutated, and so on and so forth. So the selection of those residues to be present inside the protein is also due to the fact that certain residues only can be able to fold the protein properly. Other residues present in other locations will not be able to. So a certain sequence of protein will only fold. If you put random sequences of amino acids, mostly it won't fold. And so nature selects for that sequence that are able to fold. Right? Now, if we talk a little bit about the quantification of the protein folding, quantification with respect to what? Mostly we are going to talk with respect to energy, right? So, the general intuitive idea would be that folded protein state is at a lower energy than, or more stable than an unfolded state, which is true, which is definitely true, because the delta G is negative when going from an unfolded to a folded state, but the negative delta G is not that negative. So the difference in energy, gives free energy between an unfolded protein state and a folded protein state of the same protein is not that drastic. Right? It is favorable. It is no doubt it is more favorable, but it is not drastic. So what happens is, in some cases, what you will see is reversible processes between folding and unfolding. So the protein, under certain contexts, if the cellular environment has certain things or certain context in terms of pH, temperature, and other kind of things, it will fold. And when that context goes away, when that environment goes away, it will become unfolded. So reversible reactions can also happen, which is not kind of characteristic of exothermic reactions where you have a negative delta G. Here we have a negative delta G, but we also have in some cases reversible processes of protein folding because of the magnitude of delta G, because it is not very high. Now, protein folding can be considered a thermodynamic transition in which the system changes between an unstructured unfolded state to a folded state. So for a single molecule, you can write from unfolded to folded. This is the delta G difference. So delta G naught is delta G F folded, G unfolded. Right? Now at every level, you can write delta G as delta H minus T delta S. So the idea is that there is a temperature dependence on the folding of the protein itself. So there is this particular cutoff at which, so this is the T fold or the TF. Below this temperature, the change in free energy upon the folding is negative. The protein folds spontaneously. Above this temperature, the change in free energy upon folding is positive, and the protein unfolds spontaneously. So as I was saying, this is a reversible reaction, right? So under certain contexts, under certain temperatures, under certain pH conditions, under certain environmental conditions, the protein will fold spontaneously. You'll have a negative delta G. But the moment you change certain things slightly in terms of the environment of the protein itself, it will go back the other way. So it will move from the folded to the unfolded state more spontaneously than from a unfolded to a folded one. Okay. So the key takeaway from here is there is a temperature dependence on the folding of the protein, and there has been several studies to identify what is a good temperature range for which protein folds positively, and there has been a lot of studies regarding that, right? Okay. Now, temperature is one way, right? So if you increase the temperature, in most cases a protein will get denatured, and it will not retain its secondary or tertiary 3D structure, and it is going to go back to the unfolded state, right? But there are other ways to denature a protein. You can unfold a protein using chemical denaturants, which includes alcohols, urea, guanidine hydrochloric acid, and so on and so forth. pH again is a contributor to that, because if you have extreme pHs, the hydrogen proton concentration basically in the cell is very different, and so those the the pH change will affect the bond formation within the secondary and tertiary structures of the proteins, because hydrogen bonds won't be as easily formed or formed in the same way as before, because now the hydrogen ion concentration inside the cell or in the environment is different. And also you can have very high salt concentrations which can denatured proteins as well, and also you can use reducing agents. So there's a host of way you can do the unfolding, right? Now this was about thermodynamics, right? So the takeaway from here is the delta G is generally negative, but there is a temperature limit to that. So if you move above a certain temperature, the delta G becomes positive. So below a certain temperature, a protein would fold uh, spontaneously, and above a certain temperature, that folded protein would unfold spontaneously. And there are other ways to do the unfolding as well. Now, if we this was the energy requirements for protein folding, the thermodynamic requirements for protein folding. But there is also the kinetic part, right? The rate at which the folding occurs. But why is protein folding kinetics important? Why would you want to study kinetics? Uh, just a second, I'm just turning off my camera. I'm having some internet issues. So yeah, please let me know if there is some technical issues, because I'm facing some internet issues today. So just to be safe. Okay, so why is protein folding important? Because folding kinetics. One second. Excuse me. Sorry. Uh, so yeah, in the early 1960s, where protein secondary structures were getting discovered, protein folding was being talked about. uh, Protein folding kinetics was central to the historical development of protein science. In 1968, this person, Cyrus Leventhal, we are going to talk about the Leventhal paradox a little bit later, posed a puzzle that is called the Leventhal paradox. Proteins face a needle in a haystack problem, right? The haystack is the astronomically large conformational space that an unfolded protein must search in order to find the needle, its one native structure. So the idea is that if you have a sequence of amino acids in a polypeptide chain, that polypeptide chain to form a protein has to fold in certain ways in an exact certain manner to form the protein of desire. But that same amino acid sequence, there is option for it to fold in certain other ways, right? So there are n number of possibilities in which a protein can be folded. But the idea is that generally uh, the, the the protein folds in a way that is already decided. So the idea is, how does the protein know to fold in this state versus not in other state? And this paradox, this finding a needle in a needle in a haystack. So the needle is your perfect structure that you want to get to. And the haystack is basically all the possible structures that your primary sequence uh, can go to, right? So uh, yeah, so this was the Leventhal paradox. We're going to discuss about this a little later as well. So if you estimate that there are between z equal to 3 and 8 conformation states for each peptide unit, and there are n equal to 100 amino acids in a position, then the number of conformations the proteins must search, which is the size of the haystack, is huge, which is like 10 to the power 50 to 10 to the power 90. The Leventhal paradox is the question of how a protein can fold to its native structure so quickly, often in microseconds to seconds, in the light of this huge search problem. So yeah. So why do we want to know what are the possibilities of a protein folding state? We know that the protein amino acid sequence is like that, and after folding the protein is formed like that. So why do we need to know, right? So it is inspired by a very important idea that if we could learn the pathways of folding, then perhaps we could infer a folding principle or mechanism by which the conformations are explored or ignored, as so many different types of proteins reach their native structures so quickly and directly. So the idea is that if you learn the pathways, learn the mechanisms by which the protein folds, then you can infer the paradox that why is it folding in a certain way and not in a certain other way. And perhaps knowledge of the folding intermediate states, there will be in a lot of cases where there are large proteins, it can form intermediate states right while folding, because uh, it can't directly go to the final stage. It will form an intermediate stage, and it will move from the intermediate state to the final stage based on the energy differences, right? So understanding the protein folding intermediates, which are partially assembled structures that are formed on the way to the native state, could give us clues about how each type of protein knows which vast stretches of its conformational space not to search. Such knowledge could help us improve computer search methods for predicting native protein structures from their amino acid sequences. And the knowledge of protein folding kinetics could also help us understand the dynamics of other processes, including ligand binding, allosteric changes in conformations, and other mechanistic actions, right? So everything that a protein does in a physical manner, the interactions that it is going through, is guided by the folding of the protein ultimately, right? So if you can determine what is the mechanism by which a protein folds, you can determine several other interactions the proteins make with, say, for example, a ligand, between a ligand and a substrate, between sorry ligand and an enzyme, between DNA and transcription factor, between allosteric changes in conformation in enzyme kinetics as well, right? So the idea is that there are a lot of possibilities a protein can fold into, but it generally folds into a native structure which is predetermined. So the paradox was that if there are like 10 to the power 50 or 10 to the power 80 possibilities, how does the protein determine that this is the most probable structure for my sequence, and that is how I'm going to fold? So what is the understanding behind that? That there is a huge search space, but the protein always folds correctly to its own native folded stage. So if you want to know why this is happening, you need to understand the mechanism behind the folding, different interactions that are driving the folding, and basically, uh, yeah. So if you have a mechanistic insight into that, you can predict different foldings from their amino acid sequences as well, right? So experimentally, how would you calculate folding rates? Because like for calculating the kinetics, you need to have some sort of experimental data which will tell you about the uh, the, the different rates at which the reactions happen, right? So if you begin with the most basic experiment, you start with a dilute solution of proteins that are unfolded because of the denaturing conditions of the solution, and then you switch the solution to folding conditions. So basically, you you first put the uh, uh, you first preserve the proteins in an unfolded state uh, with some denaturing agents, and then you take the protein out and switch the solution to a folding conditions by suddenly changing the denaturing concentration, pH, or temperature. You then observe an optical property, which is which can be either fluorescence or circular dichroism, where uh, yt will be a function of time t. So in this case, the left yt is a function of time t, and you monitor the rate of the increasing population of native molecules in the test tube. This type of experiment is also called in vitro refolding. So what you're doing is you're trying to fold the proteins in an external condition which is mimicking cellular chemical concentrations, right? Uh, so this type of experiment is often called in vitro refolding because it takes place in test tubes, not inside the cell, with proteins that were initially unfolded. You can also run the experiment in reverse, unfolding native proteins by jumping the protein solution from native conditions to unfolded conditions. So you can go vice versa. You can go from an unfolded to a folded state, and you can come back from a folded to an unfolded state, right? So, for the folding and unfolding are generally well described by one or more exponential functions of time. So this is the first order kinetics, if you remember. So the protein folding follows a first order kinetic equation, and this is the equation by which it is given to. If you need to solve this, you need to use some linear algebra concepts, get the eigenvectors and the eigenvalues, and you can solve it from there, right? So these plots actually plot the yt, which is the folding rate, with respect to time. So these are here for three different enzymes or proteins. So this section, if the protein is in somewhere this section, it is unfolding. If it is somewhere this section, it is folding, and so similarly for the other proteins as well. So yeah, so this is a log plot, by the way. So this is log yt versus time, and this is just log yt versus time. So these are logarithmic plots. So if you try to plot this in a normal plot, this would look like exponential, right? So kinetic relaxation of the amount of native or denatured protein as a function of time after jumping the conditions by light absorbance for lysozyme, the first one, cytochrome C, the second one, and ribonuclease A, the third one. A single exponential is a straight line on these logarithmic plots, and these plots show two-state folding and unfolded kinetics for lysozymes, three-state folding and unfolding for cytochrome C, and a combination of three-state and a sequential folding of ribonuclease A. We are going to talk about two-state and multi-state folding in the later slides as well. Okay, sorry. So if you have a typical single small domain protein, it will fold or unfold in just a single exponential relaxation process, such as the one shown in the figure for lysozyme. So it will go from directly from so it does not go into any intermediate stage. It goes from the unfolded to the folded or vice versa state. Right? So processes in nature that involve single exponential dynamics are said to follow two-state kinetics. Because the mass action model you need to explain them requires only two states, in this case the denatured and the native state. So it is a reversible reaction. It is going from denatured to native with a forward reaction rate of Kf, and it is coming from native to denatured with the constant of Ku. So this is a reversible reaction. So where your Kf is the folding rate coefficient and Ku is the unfolding rate coefficient. The time dependence of this process can be written in this manner, which is of a first order ODE. And when D and N concentrations are time dependent concentrations of the denatured and native states respectively. If you choose the normalized counts, then D and N are time dependent probabilities of populations, and PDT and PNT are values ranges from 1 to n. So basically you can replace the D here with some probabilistic function, some distribution from which it can sample randomly between 0 to 1, right? So on the right hand side are the different other methods or other models for folding. So the first one is a two-state. So it is either residing in an unfolded state or it is residing in a folded state. There is nothing much in between, right? So it is directly going from a denatured to a folded state. Then you can have intermediate in when you're moving from a denatured to a folded state. You can have stable intermediates which are basically forming because of certain interactions inside the protein, right? So you can have three state with on path intermediate as well. In this case I is the intermediate. You can have three state with off path intermediate as well. So it is directly forming an unfolded protein from the unfolded protein, but while forming this it is also forming some other intermediate which is not which is not kind of related to the folded protein itself. So it can also be possible that you are seeing isomers. So this protein fold different folded proteins of the same protein will be structural isomers of each other, right? So it can form different isomers. So in the third case, what it will form is two different isomers of the same protein folded in two different manners. And in the last case is basically you can have multiple intermediates before reaching the folded state from a denatured state, right? So yeah, a lot of possibilities are there for in case of protein folding kinetics, but the idea is that generally um a protein would like to move in terms of its energy. It would like to move from a folded to sorry, from an unfolded to a folded state. Sorry, I thought something else. Okay. Okay. Now, what are the properties of proteins that are influencing the rate of the folding, right? So first is the sequence of the protein itself, as we have discussed earlier and everything. The sequence of the protein is the single most important thing for the protein, right? So the amino acid makeup of the protein is very important. Just a second, I'll be back in a minute. Okay. Yeah, sorry for that. Okay. Yeah, as I was saying, the again the sequence of the amino acids in the protein will determine how the protein will fold, right? So based on the amino acids, you can form categories of the types of amino acids that fold fast or slow or in an intermediate manner, or like whatever. Like depending on the amino acid residue, you can determine if it will be a fast folding protein or a slow folding protein. So in fast folding proteins, it will be dominated by polar residues like lysine, serine, threonine, whatever, right? There's a reason for that. Can you, if you try to understand why would polar residues be more in fast folding proteins compared to slow folding proteins? So if you have polar residues in your protein, so if you have polar residues, you can form those salt bridges, you can form ionic interactions, which are much stronger and more easily formed than hydrophobic interactions, right? So if you have a lot of your polar residues in proteins, they are supposed to, they are generally going to fold faster compared to if you have slow folding proteins. You'll see higher concentrations of hydrophobic residues like alanine, cysteine, glycine, and leucine, right? So hydrophobic core formation involves long range interactions which will take longer to establish than polar short range contacts, because if you have polar residues, you can form easily ionic bonds, salt bridges, and in a very close proximity they can be easily and in, formed in a fast manner. But hydrophobic interactions uh, are ones that take longer to form, longer to establish than polar short range contacts. So that was the sequence of the amino acids. So the type of residues that are present will determine how fast the protein will fold. Now the second point is the topological parameters. So in case of this, we quantify a parameter called contact order. So contact order measures the relative importance of local versus non-local contacts. So this is the contact order formula. So if you have so it's on the numerator you have the sequence separation, the length of sorry, the number of residues between uh, any two residues that you are trying to find the contact order of. So delta S will be the distance between those two residues, L will be the total number of residues, N will be the total number of contacts, and if you sum them up over different pairs of residues, you find the contact order. So if you have high contact order, it means many long range interactions are slow folding, hence low contact order indicates fast folding. So if you have high contact orders, there is it indicates uh, these are long range interactions. So these are basically hinting towards hydrophobic interactions. So if you have higher value of contact order, it means there are long range interactions, and those are driving hydrophobic interactions. So it will fold slower compared to faster when you have lower contact order values. And the third thing is basically what we were saying, long range order. We have discussed this in quite details in earlier classes. So basically, it is defined as the contacts which are closed in space, but they are far away in terms of the distance between the two residues. So two residues can be away for a very so be, so two residues can be separated by a by a large length of residues between them, but they can come into contact with each other because of the folding of the protein, and so these are known as long range order or long range contacts, right? So if there is a long range contact, so if you look at the data, if you quantify the long range order of the available proteins, and then you see and then you try to uh, plot that with respect to the folding rate of the proteins. So LRO will always show an inverse relationship with experimental folding rate. So higher your long range contacts, slower is your protein folding, which is also intuitive from the first preference as well. So if you have hydrophobic residues, uh, it means if you have more hydrophobic residues, what it means is that you'll have more hydrophobic interactions, and which will delay the folding rate, and it will slow down the folding rate. Now if you have high LROs, high long range orders, what it means that you have uh, you have mostly higher number of um, hydrophobic residues present inside, because the folding has happened in such a way that the polar residues has come together, and in between the fold there are all your hydrophobic residues. So this also means that your hydrophobic residues are also higher, which also intuitively will tell you that the protein folding rate will go down. So if you look at the data for long range order and also for another parameter called multiple contact index, it shows negative correlation and inverse relationship basically. So uh, and also LRO, if you have high long range order contacts, it is particularly a very good predictor for all beta proteins, all the beta barrels and all those kind of proteins. Those It is a very good predictor for LRO is a very good predictor for that kind of folding as well. So there is this parameter as well, which is the multiple contact index, and it identifies residues that form multiple contacts with other residues. And if you have a protein with higher multiple contact index, it is generally supposed to fold more slowly. And if you see the data, you'll find the correlation that it is quite negatively correlated with the protein folding rate, but it forms a better correlation with folding rates than standard contact order for two state proteins. So contact order is something on which the long range order and the NCI has been built on, and these are better predictors of the folding rate rather than contact order itself, right? moving on. So now we come to a very interesting theorem called Anfinson's theorem, right? So environmental factors. So this person, first let me tell you, this person won the Nobel Prize for this particular work. So what did he do? So the background is that environmental factors such as solvent properties, temperature, and pH are known to contribute to the specific 3D structures of a native protein. As we have discussed, if you change the pH or change the temperature or change any other uh, parameter for protein, any other kind of parameter, it will change the spontaneity between folded and unfolded protein. So the rate of protein folding will change based on these other parameters: solvent properties, temperature, pH, etc., etc. Right. However, the most important determinant of the folded structure is the amino acid sequence itself. As I was saying from the very first slide, the most all of these interactions are very important, but the one most determining thing about how a protein will fold and the rate at which the protein will fold is the sequence of the amino acids making up that protein, right? So this person, Anfinson, showed in his Nobel Prize winning renaturation experiments that proteins can be denatured and then will spontaneously refold to their native forms when conditions are changed back. So for example, if you have in a cell, what you do is you increase the pH drastically, you increase the temperature of the medium as well. So what will happen is the proteins will denature immediately. But what he showed that is if you put the conditions back, if you pull it back to the same pH and the same similar kind of temperature, it the proteins generally tend to spontaneously refold to their native forms. So these findings, so this is a quite an interesting finding. So basically, if you just change these external factors to a certain thing, you can change the folded protein to an unfolded protein, and if you bring it back to its native conditions, it will come back to its native state of folding as well. So these findings resulted in general acceptance of what is now called the thermodynamic hypothesis, which states that the folded structure of a protein is fully encoded by its sequence, and the protein finds this structure due to thermodynamics laws and obviously due to evolution. We can rephrase the idea behind this theorem by saying that the folded state is most likely the lowest free state in the native conditions. For this to hold there are three important conditions. The first is the uniqueness of the free energy minimum given the sequence. Second is the stability of the free energy minimum, and third is the kinetic accessibility of the free energy minimum. So for the first case, what it is asking for is the uniqueness of the free energy medium given the sequence. So what it is suggesting is that a naturally evolved protein sequence folds specifically to a specific structure. In other words, the sequence is the recipe for the exact structure the protein will take, right? So so it is unique in that sense. In case of the third point, which is the kinetic accessibility of the free energy minimum, it suggests that the folding and unfolding rates are sufficiently high, and in other words, that the barrier between the unfolded and the native state should not be so high, right? Uh, High barriers may, in practice, prevent a protein from reaching the folded state, because if you remember, in the delta G graph, there, if you move from one state to another, you have to cross the activation energy barrier, right? So to do that, the protein is also moving from an unfolded to folded state, so it has to also cross that activation energy barrier. But if the barrier is too high, it does not work well. So it can't be too high. So if it is too high, the protein can't go into the folded state. So high barriers may in practice prevent the protein from reaching the folded state. Note that some proteins may require special conditions to fold as well. Although thermodynamic hypotheses seem to hold true for most of the naturally evolved proteins, there are many proteins where the functional state which observed in nature is not the one with the lowest energy state measured in vitro in experiments. So the last line is the one that you should be focusing on, is that although the thermodynamic hypothesis is telling you that for most So for all proteins, the folded state is a more stable state than the unfolded state, and there can be like thousands and thousands of these folded states. But there are proteins where the functional state, which is observed in nature, is not at the fold in which it has the lowest energy. So it chooses which state it wants to be in, and it is not necessarily the lowest energy state given that particular protein, because you can find exceptions to that rule, and you can find in vitro proteins inside the cells which are not in their like lowest energy states as well. So this is the work that won the Nobel Prize in I think 1970s, and this is what is known as Anfinsen's theorem. Okay. Okay, now we come to different types of protein interactions, right? So, as we all know, proteins form highly specific and stable complexes with other proteins and other ligands. And protein interactions mediate vast majority of biological processes. So, yeah, that is why we are so interested in understanding the structure of the protein, because interactions of between proteins, or interactions between protein and DNA, protein and RNA, protein and some other organelle, uh, it happens because of the structure of the protein. It is the structure of the protein is what facilitates that interaction, right? So it can be a transcription factor binding to a DNA. It can be a cell surface receptor ligand interaction as well. So yeah, so they come in many sizes, shapes, chemical and energetic content which are unique to each interaction. So although we are getting better at predicting the structure from sequence, an elusive goal remains to predict interactions, protein interactions in the structure. So this is some of it has been looked at by AlphaFold, but there's a lot of it still not understood. Ki- Can you predict the interactions of the proteins that are going to be amino acids that are going to be present, right? And despite the diverse biological origins, all protein interactions are mediated by a common set of physical properties. At microscopic level, protein binding and enzyme catalysis uh, can be described by the same underlying chemical properties as protein folding. Binding proteins have evolved to form stable, long-lived interactions with partners, and enzymes have evolved to form transient interactions to enable rapid turnover. However, some of the microscopic descriptors differ. Right? Okay. So, uh, we know that there are a lot of interactions of proteins, different amino acids going on, and also between proteins and proteins. So how do you identify the interface residues? So what are these? So these are specific amino acids that physically touch or interact with the binding partner. So if you have a cell receptor, there is a part of the receptor to which the ligand comes and bind, and that part is the only part that interacts with the binding partner, right? So you can assume to be that. There are three major ways to identify them. First is the distance based method, which is quite straightforward. You just calculate Euclidean distances. Uh, so if you have any heavy atom or of a residue within 5 angstrom of the binding partner, it is an interface residue. So C alpha and C beta atoms often use a 6 angstrom cutoff, and the distance is calculated by this simple Euclidean formula: x minus x1 whole square, y1 minus y2 whole square, and z2 minus z1 whole square. Right? So yeah, yeah. So we now talk about solvent accessibility method. So this is another method by which you can you can quantify protein folding, right? So excuse the curly brackets and everything. So I've used this from latex, so it has directly copied the latex text, which is not be which is not interpretable in PowerPoint. So apologies for that. Okay. Uh, so when two proteins bind, the surface area exposed to the solvent decreases because the interface become buried, right? So a residue is part of excuse me. Uh, excuse me. Okay. So a residue is part of the interface if the difference in its accessible surface area between the free state and the bound state is greater than zero point 0.1, right? And there is another energy based method. So this is the solvent based method, and the other energy based method is that it looks at the physics of the bond rather than just the geometry, and it looks at force fields appearing from different types of bonds, including van der Waals forces, hydrogen forces, and other things. So there is this tool called Lambert 5 force field, which you can use to calculate the interaction energy, including van der Waals and electrostatic forces. And a common cutoff is chosen as to be, I think, -1. The latex code is not working here. Residues with lower energy, lower uh, than this, are considered binding residues. So what you can do is basically you can quantify each of the residue. So why are we doing this? Why are we studying so many interface residues, solvent accessibility? Why are we studying so much energies and whatever is going on through that? So that you can predict the folding, right? So if you can find each of the residues and you can quantify the residues based on their contact orders, their distance criteria, their solvent accessibility, their energies, and all of those things, you can put an ensemble of this together and predict how the protein is going to be folding, right? Right. So all of these methods are used for computational prediction for the protein folding, right? So this is another parameter that we use to predict, which is interaction propensity and pair reference, right? So not all amino acids are equally likely to be at a binding surface, right? So if it is a binding surface, there are specific residues that are specific to specific functions. So in this case, binding interface, there will be specific residues there. So you can calculate propensity of for binding, and this value will represent the preference of a residue type to be at the interface, and it is calculated as total number of binding residues by total number of residues. So this will tell you the fraction of binding residues that are present inside your in your protein, right? So in case of residue pairs, some pairs are highly preferred. Say for example, arginine and aspartic acid often pair up to form salt bridges for electrostatic interactions, and there can also be hotspots. So there are specific residues where a mutation causes a significant drop in binding affinity, where the delta G. So basically delta G is the unfolded minus folded, and this is like between the mutated and versus non-mutated. So delta G will be greater than 2 kilocalorie per mole. So mutations can occur which can significantly disturb the protein folding, because it, it, it brings about a drop in the binding affinity of the amino acid residues there as well, right? So so many things, but what we have gone through today, but all of these things can be implemented in certain tools and softwares. So there are three examples that has been given here. So the first one is the PDB sum, which is a pictorial database that provides a visual summary of interactions, including schematic diagrams of hydrogen bonds and salt bridges. So PDB sum is one of the most widely used. Then you have Proximate, which is a database containing thermodynamic data of protein-protein binding, specifically tracking how mutations affect binding affinity, and it gives you the delta G for over 6,000 mutant in proteins. Right now the numbers will be higher. These are like old numbers. And the last one is BINT, which is another related database which can tell you about protein-protein interaction thermodynamic data. So protein-protein interaction and what is the delta G associated with that. So for that, PINT is the database to look for. Right. So finally, we come to a little bit. We'll touch about drug design, right? So we are talking about secondary, tertiary structures of proteins, protein folding, generally a 3D structure, how a protein can go from a polypeptide chain to a 3D structure, right? Now, where does drug design come into all this, right? So the idea is that what is drug design? Drug design is basically you design a molecule which you use to target some other molecule. So for example, you can use, so there are receptors inside cells, right, which bind to certain ligands and do certain metabolic activity, not metabolic, certain cellular activities, right? So there are receptors in the cell which can do that. Now if that receptor substrate binding does not happen, or there is some mutation in that so that the binding is not happening properly, so the cellular processes will be hampered and specific diseases can be formed as well. So in case of drug designing, what you do is you specifically design a protein, specific, in most cases it is a protein, which can structurally go and bind to another protein of interest for your case, right? So what you want to do is want to predict at the very basic level, you want to predict the structure of the protein that you want to use as a drug, and if that structure actually binds go and goes and binds to the ligand, sorry, to the receptor properly or not. So you can computationally and also experimentally prove that this is a good design for a drug to go and bind to this particular receptor, or this is not, right? So that is the general idea of drug discovery and how it relates to protein folding and biophysics in general, right? So historically drugs were discovered accidentally. So we had natural sources for these drugs. So you had quinine for malaria, you had other drugs, naturally occurring drugs for different types of diseases. So since the 1960s, since we are getting to know the structure of the proteins better, structural biology has allowed for the deliberate design of enzyme inhibitors. So if you want to inhibit a particular enzyme, what you need to do is design your drug in such a way that it mimics the substrate of that enzyme, so that competitive inhibition can occur, so that your drug, when a person has eaten, will competitively inhibit the actual substrate from going and binding to the enzyme. So that is what is the deliberate design of enzyme inhibitors. So mm, yeah, so how is the ligand supposed to basic what are the basic properties of the ligand that is supposed to be your drug, right? So so the ligand is basically defined as the molecule that binds specifically and reversibly to the target protein to form a complex. So in this case, the ligand will be your drug. So the ligand binding will alter the protein's 3D shape, which will change its functional state, and the parameter that is used to quantify uh, the attraction between the ligand and the receptor is called affinity. So this is the term used to describe the protein ligand interaction, right? So now we have something called Lipinski's rule of 5. So in case of drug designing or computer assisted drug designing, whatever, there are like 5 rules that this person proposed in distinguishing between drug like and non drug like molecules, right? So it predicts high probability of success or failure due to drug likeness for molecules complying with 2 or more of the following rules. So the first rule is that molecular mass should be less than 500 Dalton. Second rule is that it should have high lipophilicity, so a lot of very high lipophilicity, and it is expressed in log P. Uh, Less than 5 hydrogen bond donors and less than 10 hydrogen bond acceptors. And molar refractivity is another index that it measures, and it should be around 40 to 130. These filters can help in early preclinical development and could avoid costly late stage preclinical and clinical failures. So basically, these are like the thumb rules that you should follow while designing the drugs. These are very basic rules that are there. After that, after you follow these, you then uh, design your drug specific to the receptor that you are designing it for. But generally these rules are followed, um, which helps in like meaning like there are less chances of failure if your molecule follow these five conditions than not, right? Okay. Yeah, now we are going to talk a little bit about the very briefly about the different interactions between ligand and receptor, or ligand and substrate, whatever you want to call it, right? Okay, so so there are two sides in the ligand. So one is the binding side, and the second is the pose active side or the active side. So basically, uh, the binding site is basically a cavity on the protein surface where the ligand will bind to. The pose is basically the geometric specific geometry, which is the location, orientation, and conformation of the ligand at the site. And significance is basically effective drugs must have sufficient interactions within the site to remain bound. So your drug needs to be designed in such a way that your that there are sufficient interactions between your drug and this pose site near the binding site of your receptor, right? Then you can use molecular docking techniques. So you can use what so you will have, like, say for example, hundreds of this drug target possibilities, and you can use molecular docking simulation techniques to find the best fit between your ligand and the protein. So there are programs which select orientations that maximize interactions while minimizing total system energy, and it will predict both the pose and the binding affinity scores. So these are mostly computational methods to do. And so based on the design of the ligand, it can be classified into if it is a structure based or it is a ligand based. So structure based uh, will be used when the target protein structure is known. You focus on docking and de novo design. So you know the protein that you are targeting, you know every detail there is to know about it, you know its structure and everything. So you can use molecular docking techniques and basically create a de novo design, create a like a fully new design from scratch and use that as a drug. And the second is ligand based design, used when the protein structure is unknown. It relies on similarity and searching. And the third option is QSAR, which is quantitative structure activity relationships, which is another technique used when many ligands are known. So yeah, so either you know the structure of the ligand or you know similarly structured ligand, and then you design your drug based on that, right? So based on continuing from that, protein and ligand preparation would be dependent on the structure preparation of the protein. So it includes removing water molecules and adding missing hydrogen atoms to the PDB file. So all we are talking about in a computational manner. So probably you do it in something called a PyMol or Autodoc Vina or something else. So you need to remove water molecules. You need to have protonation, which is correct assignment of protonation states, specifically for certain amino acids for docking accuracy, and also ligand torsion, which is basically during docking both bond lengths are fixed, but the torsion angles are changed to explore different shapes. So yeah, so basically, if you have if you have a known structure, you make your drug or your protein based on the structure known. If you do not know the structure, you base it on something similarly, um, which have a similar sequence, and then you prepare the protein and the ligand. So basically, you need to remove all the water molecules because you are going to when the interaction occurs, the water molecules will be released, right? So to remove the water molecules from from beginning, and basically you can change with you can play around with the hydrogen bond formation, which is the protonation, and also ligand torsion, which is basically the twist of the angles between different bonds, because the bond lengths are fixed, but the torsion angles can be changed, and you can find which suits best. So all of these things you are going to be basing on the binding affinity score or some other parameter that is predicting how well your ligand is going and binding to the receptor and how good the binding is from a computational perspective. So all of these things are going to tell you ultimately how well the binding is going to occur or not, right? So yeah, I think that's all from in terms of the uh, topics I wanted to discuss. If there's any question, I'll take that, or else we'll move on to the assignment, previous assignment questions. Okay, then we'll start with the questions only. Okay. Excuse me. Do we have any answers? Four. Okay, I think you're saying D. Okay. We'll wait a few more minutes for the others to do. I'll just be back in a minute. Okay. Uh, Only two answers. Okay. One more minute. I'll wait. Okay. I'll discuss the answer then. Okay. So you're given temperature is 300 Kelvin. You're given the fluorescence upon denaturation is 256 units, and the intensity corresponding to the unfolded and the folded states are 51 and 366 units, and you are asked to find delta G. Right? Okay. So first you need to find the equilibrium constant. What is the K, right? So that would be Fu by forward by the so going from the unfolded to the folded, from and divided by the folded to the unfolded, right? So this you need to calculate first. So for this you need to the uh, calculate the individual rates, right? So this would be the forward reaction would be observed minus unfolded by folded minus unfolded. So this is that. So this comes to be 205 by 315, and for the FU as well, folded minus observed, folded minus unfolded, right? So you do that, so you get this thing. So now from FU and FF you calculate the equilibrium constant, which is the K. If you do if you divide it, it will come to 0.54. Now you know the formula for delta G, which is negative RT ln K. So K you substitute, R you know, T it is given as 300 Kelvin. So you calculate this, this comes out to be around 367 uh, uh, 367 calorie per mole, right? So the question is is a little bit tricky, because you need to find the forward and the backward rates. From there you need to find the equilibrium constant, and from once you get the equilibrium constant you can find the Gibbs free energy. So a little bit twisted, but yeah, I think someone answered it correct with option 4, but someone answered I think option 1, which was not correct. So please take a look at the solution, the people who didn't get the answer. So, and if you have any questions, please ask. Okay. Okay, then moving on to the next one. Just a second. I'm unmuted, right? Okay. The next questions are mostly easy. I'm going to wait a couple minutes for each one and then discuss those. Here I see a couple of A's. Okay. Yeah, the first one is the correct option. So yeah, not much to explain here, but okay. Yeah, the one thing is that protein with low contact order tend to fold faster than with high contact order. So if you have high contact order, mostly it is going to be uh, more hydrophobic in nature and basically fold slower. Okay. Excuse me. Yeah, the answer to this is the hydrophobic force, right? It is the most dominant. Sorry, it is the most dominant uh, parameter for protein folding. So the idea that the tendency of nonpolar amino acid side chains to bury themselves in the interior of the protein to minimize contact with the water is the primary driving force that stabilizes the 3D folded state of globular proteins. So yeah, hydrophobic effect is the interaction is the most important one. Moving on. Yeah, that is true. I don't need to explain this. So amino acid composition is only based on the sequence, but rest of them are all based on the structure. So, okay, moving on. one more minute. Okay. The answer is uh, uh, 1 second. Yeah, I don't. I think this question is a little bit problematic. So
Sir, because B or C are the answer, but I don't think so. A is the correct
no,
one.
is definitely not the one. B and C both look correct. Yeah,
Yes, sir. That's why I was confused between B or D, or because D comprises of all of the above, and B is
yeah,
how we measure the
yeah. I think
protein folding.
both the options B and C are. No,
Okay,
no,
sir.
no. 1 second, 1 second. The option B is how fast or slow a protein can unfold. So I don't think it tells you the denaturation rate. The folding rate is based on when you go from a unfolded state to a folded state, right? Not the vice versa one. The denaturation part is not protein folding rate. There is no do not we do not calculate protein folding rate on the denaturation part. So C is the more correct option to mark than B, because B is kind of telling the opposite, the denaturation part. Protein folding rate will not tell you anything about how fast a protein can be denatured in the presence of some chemical or some physical deterrents. It will only tell you about how fast a protein can go from an unfolded to a folded state based on the number of folds per second, but it is not the other way around. So option C is the more correct one. B, if it would have been how fast or slow a protein can fold, then both of them would have been correct. But I think in this case, C is a better option. Yeah, I did not see
Okay, sir.
the option B that carefully. Yeah, read the options carefully as well. It's I also did not notice that. Yeah, C is a more correct option here. Okay. Yeah, so how quickly an unstructured polypeptide chain navigates the energy landscape to reach a stable 3D conformation, not the other way around. So, yeah, okay, sorry. Okay, C1A. Yeah. Yeah, I've already shown the answer, but anyway, the first option is the correct one. So they are the interfaces are always going to be highly concentrated with your electrostatic interactions, which will mean polar amino acids needs to be part of that interface. So asparagine arginine is a very good pair because one is negatively charged, one is positively charged, so it forms a very good salt bridge. So yeah, it is ideal for interface. Okay, I see a couple of ways. I'll wait another minute. Okay, yeah, option A is the correct option. So I think I've not shown what a chevron plot is, but it is basically um, on the x-axis it will represent the distance with respect to one residue, a particular residue. It can be a center. The y-axis will determine the kinetics, right? So let me just quickly show you. Yeah. So this is the rate, and this is the okay. It can be concentration of the DNAG as well. It can be position of the amino acid also you can put in here. But yeah, so with the at this particular concentration, the folding rate reaches its minima and then it increases again. So this point is something that a lot of people are interested in, which is so this characteristic V curve is a protein folding curve. Yeah. Okay. Mm.
Sir, I'm not able to see your screen.
Oh, really? Can you see my screen now?
I don't know if it's just me, but I cannot. I think I should leave the meeting and then rejoin.
Can anybody else confirm that my screen is visible or not? Oh, really? Oh, 1 second. I'll stop and start sharing. Can you see it now?
Yes, sir.
Okay. Yeah, I was just showing the chevron plot here. So this characteristic V curve that we get, and here is the denaturation concentration, and this is the rate. So there is, at certain concentration it reaches the minima. So that is the denaturation midpoint that we are interested in. A lot of uh, yeah. So quantification of the protein folding rate is also done in terms of the denaturation midpoint, which is the CM. Okay, so this is what a chevron plot looks like. Okay, moving on. Again, it is not visible.
Sir, I think it's hung up. It's showing chevron plot only. I think so. You're on another slide, if I'm wrong.
Yeah, 1 second.
Uh, it's showing chevron plot. I was confused and I was typing. Can you see the screen?
I don't know what is happening today. Uh, can you see it now?
Yes, sir.
Okay. 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 You can still see the screen, right?
Yes, sir.
Okay. Okay. Uh, the answer is somebody I think wrote, yeah, uh, all of the above is certainly not the answer. The answer is basically distance based. So um, you use distance based metric, which is the, let me go back here, the Euclidean distance. When you try to identify interface residues, what you do is basically standard geometric Euclidean method to to find distance um, between your residue and if any other heavy atom residue is present or not. So the the C alpha and the C beta atoms, from there, uh, generally the distances are calculated, and they use a 6 angstrom cutoff. So you use a Euclidean distance metric to calculate this. Okay, so this will be distance based. Yeah. So I think Rahul is asking with respect to the last question. It's protein complex structure. So how would you do alignment on a structure? You can do alignment on a sequence, right? If you do alignment, you're correct. If you do alignment on a sequence, the conserved regions are mostly your binding motif structures and motif regions and whatever and whatnot. That is fine, but that can be only done on at the sequence level, right? Not at a structural level. So at a structural level, you cannot do alignment. So there you calculate the distances. So I hope that distinction is clear, Rahul.
Yes.
Yeah. Okay. Yeah. So this is the database PDB-sum. It tells you interactions between residues and their interfaces. Yeah. It, it gives you a visual representation as well, I think. Yeah. Yeah. So I think all of us are familiar with affinity. So yeah. So binding affinity is the term that is used, which quantitatively measures the strength of interaction between the protein and its ligand. So, and usually it's the dissociation constant for the irreversible reaction for the enzyme kinetics. So yeah, this is known as binding affinity. So yeah, that's all from my side for today. If you have any questions, please ask. Else we close the session.
So, sir, since there is only a month left for the exam,
Okay.
what pattern would will we will be expected to
I think it's
what
mostly,
pattern of
yeah,
question what pattern section A B C what
okay. I think I have to take a look and get back to you. But from what I remember is like it's mostly MCQ. You can have a few of your numerical questions, like as in like you have to type the numerical answer in a given box, or rather than all of them are going to be MCQs. It's what I remember from earlier courses of earlier years. I have to confirm once what will be the question pattern. I do not have access to that, so I'll probably write. I'll ask someone like some any TA from sir's team if he or she can update me on that, and I'll get back to you. But mostly it is going to be uh, multiple choice questions, and the weightage of the questions I don't know, like from which week there will be more weightage or from which week there will be less weightage. That information I don't think will be available, but yeah. So from what I know is this, but if you want more information, I have to find out.
Okay, so it would be helpful if
Yeah,
you
yeah. Okay, okay,
discussed
okay.
the paper pattern in one class.
Okay,
Mhm.
anyway, after the 12th week, there will be one session where we'll be discussing everything that has been covered in this course. So,
Okay, sir.
so probably that day,
So based,
yeah,
based on I think so. The based on the week completion. So on after week 12th week, we have 1 week to prepare for the exam.
yeah. So I think this is what week is going on for you. Is it like week 8 or week 9?
Week 9.
So week 9, right? So week on the week 12th, we'll have a week 12 wala live session, which will be this, and a day or two after the 12th live session, we'll have that extra session. So you'll have your
Okay,
1 week to
sir.
prepare. Don't worry about that.
Okay, sir. Thank you so much, sir.
Yeah. Okay then. There are no more questions. I'll close the session for today. Okay, guys. Thank you for joining. See you next week.
Thank you, sir.